For my biology presentation, I've decided to explore the world of fungi and specifically the delicious oyster mushroom. The ancestor of fungi was an aquatic, single-celled flagellated protist. The modern-day protists that are the most closely related to fungi are the nucleariids. Scientists now recognize that fungi and animals are more closely related to each other than either group is to plants or most other eukaryotes. Using molecular clock analysis, scientists have estimated that the ancestors of animals and fungi diverged into separate lineages between 1 and 1.5 billion years ago. There are 7 major groups of fungi: Cryptomycota, Microsporidia, Chytridiomycota, Zygomycota, Glomeromycota, Ascomycota, and Basidiomycota. Oyster mushrooms belong to the phylum Basidiomycota, which is the most complex and evolutionarily advanced of the fungi groups. This phylum is also extremely diverse, including guild mushrooms, bracket fungi, puffballs, and many more. Basidiomycota constitutes the majority of the edible fungi on the planet and are some of the oldest and largest organisms on the Earth. One individual fungi covered 15 hectares and weighed 10,000 kilograms. It was 1,500 years old. A colony of fungi in Oregon covered 965 hectares and was estimated to be between 1,900 and 8,600 years old. Oyster mushrooms, or Pleurotus Ostreatus, are a monophyletic group descended from a common ancestor. Pleurotus means sideways and describes how the stem of the mushroom commonly grows out from the side of the trees or logs. Ostreatus refers to the shape of the fruiting body, specifically the oyster shape of the cap. This mushroom is an edible guild mushroom and is the second most cultivated mushroom in the world. It is commonly found in the North Temperate Zone throughout most of Asia, Europe, and in North America, although not in the Pacific Northwest of the US. There is a lot of phenotypic variability across wide geographic ranges as well. The bodies of fungi typically form a network of tiny filaments called hyphae. Hyphae consist of tubular cell walls surrounding the plasma membrane and cytoplasm of the cells. These cell walls are strengthened by chitin, a strong but flexible polysaccharide. Fungal hyphae form an interwoven mass called a mycelium that infiltrates the material substrate on which the fungus feeds. The structure of a mycelium maximizes its surface-to-volume ratio, making feeding very efficient. Fungi are heterotrophs that feed by absorption. A fungus absorbs nutrients from the environment outside of its body. Many fungi do this by secreting hydrolytic enzymes into their surroundings. These enzymes break down complex molecules into smaller organic compounds that the fungi can then absorb into their bodies and use. Basidiomycetes are important decomposers of wood and other plant material. Of all the fungi, certain basidiomycetes, like oyster mushrooms, are the best at decomposing the complex polymer lignin, an abundant component of wood. Oyster mushrooms are saprophytic detrivores decomposers, which means they break down organic material. They are particularly good at breaking down the cellulose and lignin found in plant cell walls. They are also nematophages. Their mycelia can catch nematodes, roundworms, by paralyzing them with a toxin, then digesting them. Nematodes are a good source of nitrogen for the oyster mushrooms. Fungi play a vital role in decomposition, carbon cycling, and bioremediation. Through decomposition, they release important nutrients and minerals back into the ecosystem in a form that is usable to plants and other organisms for growth and survival. Fungi are used industrially for bioremediation purposes. They are used to decontaminate the environment by removing toxins. Essentially, they're environmentally friendly waste disposal. As such, the commercial fungi growing industry addresses 2 of the major challenges faced by today's growing population. It provides food with high nutritional value and also reduces waste and environmental pollution. Oyster mushrooms reproduce both asexually and sexually. In asexual reproduction, the mycelium produces asexual spore-producing structures. After spores are produced, they germinate and form new hyphae that grow into a mycelium. In sexual reproduction, 2 mycelium of different mating types meet in a process called plasmogamy. Their cytoplasm fuses. At this stage, each cell is dikaryotic, which means that each cell has 2 nuclei, one from each parent mycelium. Environmental cues such as water and temperature change can signal the mycelia to form a basidiocarp. A basidiocarp is what you would recognize as a mushroom. Underneath the protection of the cap of the mushroom, or basidiocarp, are gills. The external surfaces of these gills are lined with basidium. It is within these basidium that a process called karyogamy occurs. This is when the dikaryotic nuclei fuse. Each basidium then produces 4 basidiospores, which will germinate and produce a new mycelium, completing the sexual life cycle. Knowing that I wanted to base my presentation on edible fungi so that I could literally eat my project, I researched mushroom growing kits online, and I chose to purchase the Pearl Oyster Mushroom Kit from Back to the Roots, an Oakland-based company that was founded in 2009. This grow kit is 100% organic and non-GMO. The substrate that the mushrooms are growing on is a block made of sawdust, wheat bran, and corn cobs. The directions that were included with the box are simple and were easy to follow. Back to the Roots also has a great program called Grow One, Give One, in which you post a photo of you growing your mushrooms to a social media platform, and Back to the Roots will donate a grow kit and learning curriculum to a school of your choice. I really enjoyed this process and was excited to see how much the mushrooms grew each day. I am especially looking forward to eating the fruiting bodies of my labor.